Welcome to the second edition of QuayCast, news, discussions and insights from Quay Pharmaceuticals, one of the fastest growing CDMOs in the world today. In this episode, Maura McArdle and Audrey Vardy share a discussion on Quay's preclinical service and expertise.
Hello everyone, my name is Maura McArdle. I work for Quay Pharma in business development, and today I want to introduce you to my colleague Audrey Vardy. I'm going to ask Audrey a few questions about our preclinical services at Quay. So Audrey, would you like to start by telling us about your role at Quay?
No problem, Maura. Thank you. My name is Audrey Vardy, as Maura said, and my role at Quay Pharma is I'm the Head of Business Development for our preclinical service. I've been with Quay Pharma for almost 18 months now, but my background is mainly in DMPK and investigative toxicology, and I've worked at a few different contract research organisations, both in operational and business development roles over the last 15 years or so.
Great, Audrey. And could you just maybe kick off by telling us what do Quay's preclinical services actually consist of?
So essentially for our preclinical formulation development, the aim really is to start with quite a simple solubility screen. But we like to do this very much in keeping with the ethos of Quay Pharma in general, really, and really use a scientific approach to our solubility screen. So it's not a one-stop shop. We base our solubility screen really on the characteristics of the API. We use any of the phys-chem parameters of the API that's already known. We like to understand a little bit about the downstream studies that are planned. For example, is there a particular species that's to be targeted, and also roughly what's the dose range that the client's looking to achieve. We'd like to know a little bit about the indication that's planned for the compound downstream. And also, obviously, the route of administration is very important in terms of tailoring the solvents and the excipients that we might want to look at. The amount of API that's available to the client is very important, and we also like to understand if there's any DMPK data available, and look at things such as like CACO2 and permeability and so on. So the solubility screen, although it sounds very simple, it's a very sort of tailored study that we would do to really optimize the solvents and the excipients that we would evaluate for the client. And really the aim of the preclinical service is to establish a simple formulation really to achieve and satisfy the current needs for the API. But it also allows us to build up a little bit of information around the solubility characteristics of the compound. If a simple formulation is not obvious from that initial screen, then there are other things that we can do and other avenues we can consider, such as looking at nanoparticles or solid dispersions and so on. But that's becoming a little bit more complex and is really not the starting place that we would consider for our preclinical services. So that, in a nutshell, really is kind of where we would start with preclinical.
Great. Could you explain where those preclinical services sit in relation to animal studies and formulation development for clinic?
Sure. So it really depends what stage the client's at, but essentially we can really kind of add value really at a number of different stages, depending, as I say, at what stage the client's at. So the preclinical services at Quay, we can set upstream of the initial PK studies, at your initial pharmacokinetic studies. If you're just starting out, in rodents. And really, at that stage, we're looking to, establish a simple formulation. Perhaps there's, some knowledge around the compound, around bioavailability. There's maybe some issues, maybe with similar compounds and so on. And so we'd be looking to, really kind of tailor the formulations to perhaps try and improve bioavailability, for example, and really try and get the best that we can out of the molecule, in terms of PK profile. And at this stage, we would really recommend that the client works with, with a DMPK provider as well, you know, and really to ensure a sort of a joined up approach for those very, you know, initial sort of PK studies. If the client may very well be a little bit further down the road, they might already have their PK studies, they may have a good handle on bioavailability, but they may be upstream of their toxicology studies and really are trying to perhaps improve exposure levels, or really just want to, you know, to optimize the formulation at this stage, just to give them the best chance of a successful toxicology study. And we would really recommend that you do some of the solubility sort of screening and excipient compatibility for these studies ahead of your dose range finding studies that you might be doing, where you're looking at a number of different dose levels over maybe like a 7-day toxicity study. And we can provide the formulations to the CRO that might be conducting these toxicology studies, or we can provide quite detailed instructions on how to prepare the formulations if they're a little bit more complex. In terms of where it sits with the more sort of formal formulation development, if at this stage when the clients are moving into their tox studies, if they're happy, then we can actually proceed, you know, forward into some more of the pre-formulation studies and into some more of the early formulation development studies, really while the GLP tox studies are really sort of coming to an end. And that's certainly something that we can do if the molecule's looking very positive and everything's looking good. Then we can kind of tailor the program to really, you know, enhance your timelines as much as we possibly can at that stage.
Okay, and how much would a typical type of preclinical project cost, and how long would that take?
So something that I haven't mentioned about the preclinical services is really around the analytics. So how we go about analysing your compound at this stage. So ideally, we would be using a plate reader type approach, really very much for sort of screening a lot of different solvents in parallel. But for this, we would need, you know, your compound to give a good absorbance over the 300 nanometers, just to avoid any interference with any of the excipients, particularly if it's more lipid-based sort of solvents and excipients that we're looking at. So provided that there's a good UV absorbance above 300 nanometers, we can use our plate reader. And this is very kind of fast turnaround, low cost, if you like, because we can assess a lot in parallel. So we'd be talking, you know, maybe £3,000 for a screening study, and we would be looking at a turnaround time of around sort of 2 to 3 weeks for that. However, if that's not possible, then we at Quay Pharma, we do have a wealth of experience of setting up HPLC analytical methodology. And for some of our clients, this is the approach that they prefer to take, or maybe need to take, because of the, you know, the lack of a decent UV absorbance. So we can easily set up an HPLC analytical method. And if we do go down this approach, we can still do the solubility screening. But if we have an HPLC analytical method, it also allows us to maybe do some short-term stability once we have maybe 2 or 3 prototypes identified from the formulation screens. This is a little bit more in depth, takes a little bit more time, obviously, to work up the method, to do the solubility screening, and also then to build in some stability. And for this, we would be looking at maybe in the order of you know, £15,000 to £17,000, and we would be looking at maybe up to 9 weeks to kind of run that. You know, but it's a much fuller package of work.
Okay, and what's the benefit to my overall project in having this work done? You know, as I move towards clinic, you know, I guess a lot of clients maybe wouldn't consider doing what you've just outlined, and you know, what's the payoff for doing that, Audrey?
Absolutely. Well, if we go down the screening only route, then we can really achieve, an improved formulation for you going into your PK studies or your toxicology studies. So, can really help to improve, as I say, either the bioavailability or the toxicology exposure that you can achieve. And we can do that, as I say, in a very short time frame, if we're going down the screening route in a couple of weeks. And we'd be looking at using maybe 500 mg of compound. So in terms of API, which at this stage in your program is obviously very precious, then a small amount of compound can be used to really gain some quite important information on the compound at this early stage. However, it won't negate the need to do, all your proper pre-formulation studies and then obviously your formulation development. However, if we go down the HPLC analytical route, then this means that at Quay Pharma, we would have your method already established. A fairly extensive solubility study would already be complete, and we would have an indication of some early stability, maybe short-term stability over maybe 7 days or 10 days. So really, by the time we would do that preclinical package, we would really have quite a good package of information regarding the API. So then when you move into your pre-formulation, our formulation scientists and technical experts would already have quite a good feel for your compound. They would then be able to use that information really to tailor, an excipient compatibility study. And from an analytical point of view, we would really just have to do the force degradation study then, and that would really kind of tie up your pre-formulation work very well. So it really could reduce your pre-formulation timelines and also the costs associated with your pre-formulation studies. So although you're investing a little bit earlier on, you really can see the benefits of that, and you would then have some cost savings a little bit later on in your program too. So yeah, it's a little bit of a win-win situation in terms of your timelines and your costs for the later part of your project.
Okay, so really the cost of the project and the immediate benefit is hopefully improved performance and results from your animal studies, but the work that you've done will actually reduce the amount of work needed to go into clinic eventually as well, potentially.
Absolutely, yeah. So as I say, win-win.
Okay, great. And how do these services, so far we've talked about how they would work for new chemical entities and new drug products, but how would these services lend themselves to reformulation projects, of existing drug products?
Yeah, so this is something we've done quite a bit of work on as well at Quay Pharma. So really our approach to the reformulation projects within the preclinical team is twofold. Firstly, we would perform a gap analysis, and from this is very much theoretical, it's very much literature based, where we would, from a formulation perspective, do an extensive literature search, look at all the information available on the API. We would build in looking at patent searches and any sort of patents or liabilities that need to be flagged. And really from all the information that's available out in the public domain, we would then put together some theoretical ways forward from a formulation perspective. On discussing this then with the client, we can then go on to tailor us again, very much in kind of screening mode, looking at sort of solubility or excipient compatibility, and we can very rapidly run through a large number of solvents and excipients. And the idea behind this is really to kind of try and identify a way forward in terms of formulation that might be a little bit different to what's already on the market, or might be tailored towards a different indication if that's the drive for the client, or a different patient population, for example, if they're looking to move more into pediatric medicine, for example. And again, it's all just sort of building up an idea around formulation feasibility, and is there a sensible way forward for this molecule, you know, that hasn't already been tried, and again, we can do this at a relatively low cost, and again using small amounts of API, although the amount of API isn't always quite so critical, you know, for a reformulation project. But again, it just allows then, if the client you know, is happy with the information provided, if there's a sensible route forward, both from a theoretical perspective and also from some initial screens, then it allows the pre-formulation work to start from a much more informed position than they might have just going into it sort of cold. So yeah, again, it's a, you know, a nice package of work for not a big investment to really kind of you know, shape the way that you might move forward with a formulation project.
Yeah, that I can see where you're coming from there. It would really help if a client had a number of different molecules it was looking to reformulate into a different dosage form, of maybe helping them select which one to work with next, or which one's got the best opportunities potentially, without spending too much time or money on something that's much more difficult, but perhaps.
Absolutely, yeah. If a client had a few choices and they were trying to go perhaps with a slightly lower hanging fruit, if you like, then this is a very kind of easy, fast, you know, and relatively inexpensive way to really try and make some choices. Just add a little bit of information to each molecule and then make some good choices moving forward.
Okay, that's great. Well, Audrey, thank you very much. That was a really good sort of outline of what's involved with all Quay's preclinical services, and hopefully the benefits that the clients can see from using those services. That's great. We'll talk again soon, no doubt.
Thank you for listening to QuayCast. Please share and follow for future episodes.